In 1961, Nirenberg and Matthaei broke part of the genetic code. They did so by making use of homopolymers, molecules which consist of a single type of nucleotide. They made the holopolymers by combining polynucleotide phosphorylase to an uracil nucleotide-only solution, which then generated RNA molecules which consist of only UUU codons. In order to figure out what amino acids UUU coded for, they set up 20 test tubes, but for the sake of simplicity, I'll be using one. Each had the requirements for translation and all 20 amino acids. However, a different amino acid was radioactively labeled in each tube. Imagine that phenolphthalein is the amino acid I labeled for this tube. After translation, the only radioactive protein was found in the test tube containing the radioactively labeled phenolphthalein, leading us to conclude that UUU coded for phenolphthalein. They did a few more experiments and found out that poly C coded for proline and poly A coded for lysine. Three years later, in 1964, Nirenberg and Leder came up with another way to break the genetic code. They decided to use ribosome-bound tRNAs. This procedure involved using short mRNAs with known codons like GUU and combining them with a mixture of ribosomes, tRNAs, and amino acids. They waited for the reaction to take place, then passed the mixture through a nitronucleose filter which prevented the tRNAs that were paired with ribosome-bounded mRNA from passing through. Finally, they examined the remains in the filter to determine which amino acids bound. In this case, valine was formed. They then repeated these steps with different short mRNAs codons in order to figure out the others.